Good morning. This is Miss Watson, room 402. I have decided to do a video recap of the lessons you guys have learned about DNA and RNA thus far, hitting only the high points. This video will not cover everything you learned, but it will hit the really important parts that you definitely need to keep with you as we move on to our activity during your block period next class. So the main objective of today is that you will be able to recap the basic information that you've learned about DNA and RNA to date. A little bit about this lesson. These lessons where I do a recording and show slides, they're designed to hit three different types of learners: an audio learner, a visual learner, and a kinesthetic or tactile learner. So this video works best if you engage in all three of these at the same time. So it involves listening to the audio, watching the slides and the diagrams, and then writing in your closed notes. Studies have shown that it activates parts of your brains and leads to better, longer lasting understanding. This is a quick 1-minute video that just kind of frames the why we need to know about this, okay, where we're going with this unit. So please enjoy.
Stated Clearly presents What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur?
I like to end it on a happy note with a dinosaur eating a dog. But that is kind of the point of this part of your lesson. How is it that this molecule of DNA codes for everything that you are, that I am, basically that all living things around us are? And we're going to get into that today.
Stated Clearly presents.
All right, so nucleotides are the building block of DNA. I want you to, there's a nucleotide in your note. Go ahead and label that nucleotide the same as the picture here. Nucleotides are made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. As you go through these notes today, you can pause the video to take time to write your notes. I'm just going to be going through the video, so don't feel like you have to keep up with me. You have control of the pace of this lesson. Nitrogenous bases come in 4 types, and those 4 types can be further divided down into 2 categories: the purines, which are adenine and guanine, and the pyrimidines, which are thymine and cytosine. So again, nitrogenous bases come in 4 types. You can just remember them as A, T, G, and C: adenine, thymine, guanine, and cytosine. The basic structure of DNA, there's really interesting stories on how the structure of DNA came about, um, or how scientists discovered the structure of DNA. But we're going to save all that because remember, we're just hitting the high points today. I heard you guys even did a poster on some of the key people involved in discovering the structure of DNA. But essentially, DNA is double-stranded. So there's 2 strands of this DNA molecule, and they're connected in the middle by these nitrogenous bases. And remember, those nitrogenous bases are A's, T's, C's, and G's. And then those 2 strands that are connected in the middle by the nitrogenous bases, they twist up to form a double helix. And that is that classic spiraling staircase structure that we see in DNA. The base pairing rule. So the base pairing rule, it applies between 2 strands of DNA, and it states that between 2 strands of DNA, A will always pair with T. So adenine will always pair with thymine, and C will always pair with G. So cytosine will always pair with guanine. And there is a helpful mnemonic that you can remember the base pairs, and that is A goes with T, just like apples grow in trees, and C goes with G, just like cars run on gas, right? Cars need gas, apples grow in trees. You can also say cars go in garages. Not everybody has a garage, and not everybody's car runs on gas. But if you can remember one of those two things, it should help you remember that A goes with T and C goes with G. So just a quick review, DNA is called DNA because it stands for deoxyribonucleic acid. It's a double-stranded molecule in the shape of a double helix, and it's composed of those repeating nucleotides. Nucleotides are made of a phosphate group, a 5-carbon sugar, which is called deoxyribose in DNA, and a nitrogenous base. Nitrogenous bases come in 4 flavors: guanine, cytosine, adenine, and thymine. And they pair together in the middle of that DNA structure. And they pair together according to the base pairing rule. A goes with T and C goes with G. Now, how are these strands held together? Well, in a single strand of DNA, and remember that DNA is double-stranded, but in that one single strand, those nucleotides, the phosphate group, the 5-carbon sugar, and the base, are held together by strong covalent bonds. Okay, these covalent bonds are very strong. They're really difficult to break. And the way that 2 nucleotides are stuck together is that the phosphate group of one nucleotide connects to the 5-carbon sugar of another nucleotide with one of those strong covalent bonds. Now, when you see covalent bonds drawn out in a structure, like the one on the right, they are often represented with a solid line. On your closed notes, you have a little diagram. You can label the covalent bonds now. So in complementary strands of DNA, remember DNA is double-stranded, and we call the strands complementary strands. We don't call them identical strands because they're not identical. So what it means is DNA is double-stranded, which means that every strand of DNA has a complementary strand running parallel and in the opposite direction. So we call this anti-parallel. The strands are anti-parallel because they are parallel, but they go in opposite directions. And the complementary strands meet in the middle with those bases, and they follow the base pairing rules. And again, that's A pairs with T and C pairs with G. Apples grow in trees and cars need gas. So on your closed notes, there's a little activity for you to fill in the complementary strand of DNA, just a little reminder of where we've been. And one more key component to the structure of DNA is that hydrogen bonds are what bonds the 2 complementary strands together. So the 2 complementary strands of DNA are held together by relatively weak hydrogen bonds. They stick together, but they're not as strong as those covalent bonds. So the nitrogenous base of one strand of DNA connects to the nitrogenous base of the complementary strand with a hydrogen bond. And you can see from the diagram, A pairs with T, C pairs with G, and so on. And hydrogen bonds, when we look at drawings of DNA, are often, but not always, shown as broken lines or dotted lines or dashes. So now we're going to start talking about DNA replication. You guys have learned about mitosis. You know that cells need to divide. So in eukaryotic cells, all of our, which are what are inside of us, those cells that have a nucleus, all of our DNA replicates or copies itself during the S phase of interphase. So our DNA copies itself during the S phase of interphase. So cells spend most of their time in interphase. There's a G phase with cell growth, an S phase with DNA replication, and a G2 phase, which is preparation for mitosis. Then there's a very short cell division phase where cells go through mitosis and cytokinesis. DNA must be duplicated before a cell can undergo mitosis, which is eukaryotic cell division. If there's no DNA replication or duplication, you can't have mitosis because you wouldn't end up with two identical cells. DNA replication is said to be semi-conservative because the identical new strands are made of one old strand, and in this drawing here, it's green, and one new strand. And in this drawing here, it is orange. And in the assignment for the closed notes, I'm going to link some helpful videos if you guys need them. But one old strand and one new strand. DNA replication is semi-conservative. For those of you that did 11.3 on DNA replication, there was a question that asked, if scientists were to have DNA in a cell and it was labeled with radioactive nitrogenous bases or radioactive nitrogen, it allowed that DNA to replicate with non-radioactive nitrogen. And excuse me, and then it underwent mitosis. It went cell division. The new cells, the daughter cells, would both of them have DNA, or would both of them have radioactive DNA? Would only one of them have radioactive DNA, or would neither of them have radioactive DNA? And if you look in the picture on the right, you can see that this green would represent the radioactive DNA. And we fast forward all the way to the far right, and now we have two sets of DNA. In mitosis, they'll be evenly distributed. So you can see that both cells would have radioactive DNA. And that is because of the semi-conservative nature of DNA, because half of the old strand and half of the new strand would be in each of those daughter cells. So the answer to that question is both cells would be radioactive. Now, we're going to talk quickly about the structure of DNA and how it relates to the function of DNA. When DNA is replicating, there's this molecule called helicase that comes through and breaks apart the double strands of DNA. And you may be thinking, how could a molecule break apart these strands? Well, the answer is the bonds that hold together those complementary strands, remember they're those relatively weak hydrogen bonds, whereas the bonds that hold the nucleotides together are those very strong covalent bonds. So helicase, which is an enzyme, can go through and break those weak hydrogen bonds, almost like pulling apart a magnet. And the nitrogenous bases will unzip in the middle and allow that DNA to be replicated. So if you look to the left of the image, you see there's two DNA copies, whereas before on the right of the image, there was just one DNA. So that is how the structure of DNA, those bonds, help DNA function and replicate. So you guys hopefully learned before that DNA contains segments called genes. And these segments, many of these segments, not all of these segments, they code for proteins, and those proteins will ultimately determine traits. And traits are any specific characteristic of an individual. So the protein, for example, melanin, determines how dark or light your skin is going to be. Melanin also determines how brown your hair is going to be. Melanin also determines the color of your eyes for some of us that have brown eyes. Okay, so melanin is a protein that is coded for by a gene and ultimately leads to a trait. Okay, so that's kind of how, in a nutshell, our DNA makes us who we are. But what if I told you DNA never leaves the protection of the nucleus? Imagine you get like a new pair of shoes for my sneakerheads out there. You buy these new pair of shoes, you keep them in their box, you put them in the closet. As long as they stay in that box and they stay in your closet, they retain their value for the most part. You can resell them at the same price or sometimes even higher than you paid for them. But if you take those shoes out of the box and you wear them around school, you wear them around the city, wherever, they start to lose value. They start to degrade, right? The bottoms get dirty. They get scuffed. They could get stepped on. Think of DNA as a brand new pair of shoes, and the nucleus of the cell is the box. Our bodies want to keep our DNA safe inside of a nucleus. So your DNA cannot leave the protection of the nucleus, but it codes for all of your traits, and your traits aren't stuck in the nucleus of the cell. So how does that happen? Like, what exactly is going on? DNA has the code for you, but it's trapped. DNA and the instructions it contains are trapped in the nucleus because it's too large to get out. But there's this fancy little molecule called RNA that can leave the nucleus easily. And it leaves the nucleus through these little openings in the nucleus called nuclear pores. Okay? So DNA is too big, but specifically mRNA is small enough to leave the nucleus, which is kind of a cool little fact. So I want you guys to take a second and read this picture. On the left, you have DNA. On the right, you have RNA. In your closed notes, I want you to write down one difference you notice between the structure of DNA on the left and RNA on the right. And remember, you can pause this video and write down what you see. All right, hopefully on the last slide, you were able to figure out the structural difference, a big structural difference between DNA and RNA. Now let's zoom in, and we're going to look at the nucleotides of DNA on the left and a nucleotide of RNA on the right. And again, I want you to figure out what is the difference between a nucleotide of DNA and a nucleotide of RNA, right? They both have phosphate, they both have 5-carbon sugar, they both have a nitrogenous base, but read that picture. What is different between these two between these two nucleotides? And take a second and write down two differences. There's two differences in your notes. Hopefully you guys were able to ascertain, love that word, the fact that there are some major differences between DNA and RNA. And so for your last activity on these closed notes, I want you to write down, kind of use that DNA, RNA both, and see if you can fill in that chart. So DNA is double-stranded. DNA has 2 strands, remember that double helix. RNA is single-stranded. DNA nucleotides, their 5-carbon sugar is deoxyribose. So it's a ribose sugar that's missing an oxygen, deoxy. And RNA has ribose sugar as its 5-carbon sugar. They both contain a 5-carbon sugar, but DNA has deoxyribose, RNA has ribose. DNA has those 4 nitrogenous bases, adenine, thymine, guanine, and cytosine, ATGC. RNA also has 4 nitrogenous bases, but it doesn't have thymine. It has adenine, uracil, guanine, and cytosine. So adenine, uracil, guanine, and cytosine. DNA is relegated to the nucleus. It cannot leave the nucleus. RNA can leave the nucleus. It's free to pop out of those nuclear pores and go where it needs to go. And then finally, RNA comes in 3 types, 3 main types. We have messenger RNA, ribosomal RNA, and transfer RNA. And those will come into play when you guys are doing your gizmos during your block period. So hopefully this was helpful. We're all caught up on DNA and RNA structure and function. And you guys are ready to attack that gizmos tomorrow. Thanks for listening. Bye.